All right, I think it is about 9:01. I realize that half of the world is asleep right now. Um, However, we are still recording, so we will be putting this will be on YouTube for people who wake up to watch it later. So I am Candace. I'm also known as Kraken on the server, and I am here to introduce to you Dr. Jennifer Hu. She is a postdoc at UCSF, and she received her PhD in bioengineering from UC Berkeley and UCSF. Is that right? So it was a joint PhD, I guess. And so she uses tissue engineering and next generation sequencing to deconstruct and reconstruct the cell-driven processes that build and maintain tissue structure. I'm personally very interested because I also do regenerative tissue engineering. So I'm very excited to hear your talk. And so Dr. Hu, when you are ready, you can take it away.
Great, thank you for the introduction. Will you enable participant screen sharing?
I apologize. It should work now.
There we go. All right, so today I'll be talking about my thesis research on controlling basically the outcomes of self-organization, the structures of memory organoids. And as Candace said, I just recently graduated with my PhD in bioengineering from the joint UC Berkeley UCSF program. So briefly about me, as that I grew up in Ohio. I did my undergraduate degree in biomedical engineering and computer science from Harvard University. And then spent the last several years finishing my bioengineering PhD, and very recently got confirmation that I will be joining a cultivated meat company called Upside Foods to use my tissue engineering research background to help create slaughter-free and more environmentally friendly real meat alternatives to conventional food. I'll briefly describe some of the ideas that come out of the Gartner Lab, because I think that it really like captures a lot of the like interdisciplinarity that we see in bioengineering, because we're trying to tackle relatively complex questions. We're building tissues in order to understand how tissues build themselves, and that involves, spatial arrangement of cells. It involves the cellular identity of those cells, what kind of molecules they're secreting, how they're talking to each other and to their environments. What's great about bioengineering is that we get to take all kinds of different methods from different fields to apply them to different parts of this problem. And so I'll be talking about the work that I did using a variety of different techniques to quantify steady state organoid structure. So I think we'll talk a little bit about structure first, because I think as people who love biology, we really appreciate how living things are filled with organized structures, that different types of cells are arranged in very precise spatial locations relative to each other, relative to their protein extracellular matrix, in order to fulfill specific organ-specific functions. And those structures arise from this, really fascinating and complicated self-organization process coming from, an initially sort of uniform-looking mass of cells that seems to spontaneously develop really lovely and complex movements of many, many cells moving around throughout this embryo. These cells are proliferating. These tissues are changing shape. These organs are forming. And of course, that makes this a very exciting process, but one that's also very complicated. So what we like to do is to focus on just a few modules of self-organization. So in my case, I'm going to be using a simplified version of a tissue to study the self-organization of the cells within a tissue. So in my case, I'm working with the mammary gland. That's the breast epithelium. And this is in the body, it's composed of this kind of large branching tree where the lobular units, the kind of bubbles, are the milk producing regions composed of milk producing luminal epithelial cells in green, or LEPs, and contractile myoepithelial cells in pink, the MEPs. And these contractile cells squeeze the milk produced by these green cells through the ducts and to the baby. So from these original tissues, we can grow these cells in a dish, expand them out, sort them out, and then recombine them into microtissues, which are much, much simpler than trying to study the tissue within its in situ context. These are organoids that are about on the order of hundreds of microns in diameter and just barely big enough to see with the naked eye. So we can start to use these tissues to ask questions about what are the ways that the cells are structured within these tissues. And when you do that, you start to realize that tissues do not look all identical. Even though these cells are able to position themselves in the proper orientation, when we say proper, that doesn't mean that they're all always the same. So there's heterogeneity or disorder, both in vivo and in vitro, that sometimes the cells are not in the right places, or they don't fully cover, the pink cells don't fully cover the green cells, or whatever. And this balance maintaining tissue structure can break down during disease progression. For example, during the transition from an in situ enclosed lesion to fully invasive breast cancer, the self-organizing properties of these cells appear to break down, and these cancerous cells are able to escape. And that kind of structural transition is something that's really quite interesting because it acts as a sort of barrier to breast cancer progression. So, for example, the Ewald lab has used live imaging of invasive breast organoids using a mouse tumor model and showed these movies of the pink cells and myoepithelial cells able to self-organize as though like a dynamic barrier against the dissemination of transformed invasive internal cells. And in only a small fraction of cases are the invasive cells able to stay at the tissue edge for long enough to disseminate. This suggests to us that the structure having these green cells at the tissue edge represents sort of this key intermediate step that's prior to and necessary for invasion. So that's sort of the background of what tissue structure is in the mammary gland and why we think that it's important. So from there, I'm just going to go and talk about what I did during my project, how I made my mammary organoids, the steady state structural distributions that I observed, and the framework that I developed in order to understand that and to control it. And to perturb it using breast cancer oncogenes. So like I described before, you can take primary tissue, grow it out into a dish, and then extract out the basic two cell types of the epithelium. And I generate microtissues by spinning them down into small microwells that are about 120 microns in diameter, little cylinders. This allows them to reaggregate. And after they've formed some nascent adhesions, you can transfer them into a supportive adhesive extracellular matrix, such as matrigel, which is a hydrogel that resembles the basement membrane seen in vivo. And so once you do that kind of thing, you can start doing some imaging, put them on the microscope, and see that these cells can start to self-organize once they're able to kind of interact with their environment and each other. And I think that the self-organizing property of cells is really quite convenient for us as tissue engineers, because we're thinking about these cells as living building blocks that have intrinsic, programmed behaviors. That means that we don't have to individually specify the locations of every single cell in the structure, right? We can sort of rely on the intrinsic self-organizing capacity of these cells to generate tissues of the desired structure. But the other flip side of self-organization is that, well, since it's kind of dynamic, cells are moving around, it seems like there's this, there's a distribution of different structures, both in the same organoid over time. So if you video the same organoid, the structure of it is changing because the cells are still moving, and across different organoids of the same cell types compositions. So what I wanted to do was understand this heterogeneity in outcomes across all of these tissues. So in order to do that, I started I had thousands and thousands of pictures of organoids, and so these 4 kind of pictures represent sort of the main outcomes that you might expect from mixing 2 colored cells together, right? You might have different core shell configurations. You might have half and half. You might have all mixed up. So I did some simple image segmentation and image processing in MATLAB to kind of get out these binarized masks that allow me to extract a lot of different morphological features. And the one that I'm going to talk about primarily as the coordinate of structure is the edge occupancy. So in this case, I'm going to basically look at the edge of the tissue and see where are the green cells, like how much of this edge is occupied by green cells or GF pre-expressing cells. So as you can imagine, the situation where all of the green cells are in the center, the way that it is like in vivo, is a low edge occupancy, whereas the opposite situation with all the green cells on the outside would be a high edge occupancy. So I can take this quantitative metric and start breaking down the organoid data into this quantitative structural coordinate and kind of start to quantify what I mean by structural heterogeneity, that I'm not seeing all of the organoids in the correct position on the very far left of this of the x-axis. I'm not seeing the peak at 0. I'm actually seeing a peak not at 0, and the peak is sort of broad. So basically these mammary tissues are self-organizing, in the right direction, in the leftward direction, but we're ending up with a variable structural outcome, despite being able to kind of do some quality control, make sure that, they're all the same size and they all have the same, ratio of green and red cells. So the incoming variability into this assay is low, but the outcomes are, generating some kind of heterogeneity. And so what I sort of realized is that there's some element of random chance to the way that tissues self-organize. And I think people sometimes use the phrase random chance to mean stuff that you can't control or predict. But actually, random chance does follow probabilistic laws. It's predictable and even quantifiable. So I'm going to illustrate it with just a simple example using some M&Ms. So green and red M&Ms. And if you took this bag of M&Ms and poured it into a bowl, I would say that you would probably not see a situation where all of the red M&Ms ended up on the outside or the inside of this bowl, right? What's much more likely, and what happens when I took these pictures and kind of swished them around, is that almost all of the time you sort of observe these more mixed up looking states. That what's happening is that because these disordered states have many more possible arrangements, it's just more numerous, the probability is higher for observing that structure. And it turns out that that idea is intimately related to the statistical mechanical property that's called entropy. So I can, furthermore, take this kind of entropy counting approach and, take it away from the M&Ms idea and just say, okay, here's this lattice, here are these different sites that are occupied by green cells or not. And I can show that the expected arrangements is the product of binomials, or looks approximately Gaussian for large numbers, with a peak at the 50-50 mark, where the cells are the most mixed up, and decreasing the most at the 0 and 1 marks at the left and the right. So those fewer arrangements, that means there's lower entropy and a lower probability of being observed. So using this approach, I can kind of put numbers on this entropy distributions for these simple lattices, but it is a little bit limited by this kind of discrete, there's this binomial stuff going on. But what I realized is that I don't actually have to take this discrete combinatorics method because I can measure structural entropy directly from the tissues in our system. Because very conveniently, I can recombine whatever cells that I want when I'm making my organoids. So I can pick MEPs that are both MEPs, but one is green and one is red. And these non-sorting organoids are going to not sort. They're just going to adopt this random jumbled configuration, just like M&Ms thrown into a bowl. And as you can see in this histogram, these structures' observed frequencies are entirely determined by that statistical likelihood or entropy as we predicted. Sorting, that is for the organoid to be observed in either the very low end or the very high end, is very entropically unfavorable. So this kind of was a very cutesy little demonstration of this idea, but these MEP-only organoids are non-sorting. So how does that help us understanding sorting? So let's go back to the abstract ideas that I introduced in the beginning, where disorder is a measure of this underlying entropy, and self-organization kind of goes towards an ordered system. It causes the system to be found not in the non-sorted state. So if the system's not self-organizing at all, you could be represented by having this flat favorability or energy function, so that no structure is more favorable than any other structure. So all of these non-sorting tissues have this flat function and adopt, as a result, the entropic or purely probabilistic distribution. But if you have self-organization, you can represent that as sort of a system where there's like an energy minimum, and that system is going to be driven to self-organize away from the entropic distribution and towards that energy minimum. And so what I can do then is say, okay, well, what are the self-organizing forces that are responsible for creating the observed distribution when I put these two different cell types together, the LEPs and MEPs, and they are able to sort? And so very curiously, it turned out that there already exists a theoretical model to describe systems like this, just not applied to cells, just typically applied to gas particles. But I hope that I'll be able to show you that indeed this framework does have usefulness towards describing multicellular tissues. And that framework is here, the Boltzmann equation. As you can see, the probability or the steady state distribution has indeed this entropy term that we described, the configurational entropy of the non-sorting system. And also these other parameters, the energy, which determines favorability, and the activity as a measure of some randomness in the system. And so this framework will allow us to predict the steady state probability distribution piece of I if we can identify basic values for these parameters. So I'm going to show you how we went about that, starting with this energy function that determines what's more or less favorable. And for the sake of time, I'm going to skip over like the decades of work in cell sorting that have sort of studied how cells use adhesion proteins and their contractile actin-myosin cytoskeletons to establish different interfaces between cells and other cells, or cells and like a substrate. So as it turns out, in a cell sorting system, the relative strengths of these interactions determines what's the most favorable outcome. So in the cells in the mammary gland, as you can see in these microscopy images, we observed that, if you just look at the cell-cell interfaces, we see that the pink cells seem to stick the most to each other. And so if you have a system where there's no ECM, the only important interactions are cells and other cells, this favors an organoid that has a MEP core that maximizes all of these pink cells touching each other. But if you put these cells in the context of a ECM environment where adhesion to the substrate is allowed, the large difference between MEP ECM and LEP ECM energies dominates and favors the pink cells to go to the outside in direct contact with the ECM. So these energy hierarchies basically drive the left or the right shifts of the observed structural distributions of organoids if we put them either in the ECM or we leave them in a suspension culture in an agarose microwell. So the observed distribution sort of looks a little bit like what we would expect given what we know about these cell energies. But in order to understand not just the minimum energy structure, but the overall energy landscape, I sort of converted these energy measurements into parameters and used those to parameterize thousands of simulated tissues of varying arrangements. So I can I can just basically permute all of the cells over and over again to make tons and tons of different tissues and add up these different relative energies to predict the total energy of the tissue. And so if I do that, I find there's a nearly linear energy gradient driving cell sorting, either in the presence of ECM or without the ECM, to minimum energy structures on the left or the right, respectively. So I'm basically taking measurements from isolated cells and using those isolated energy measurements to predict how those cells are going to act in the multicellular organoid when I put them all back together. But we can actually approach this question of the energy landscape from the opposite direction. So start with the multicellular organoids, which I can observe, and then ask what's the energy function that could be responsible for what we observe. And to do that, we can use this Boltzmann equation. We have the term, P, the probability, and the term W for the number of ways we can arrange these cells. What if I just rearrange the terms a little bit? Let's put our two knowns on one side, and on the other side, we'll have this energy function. And so if we do this, we can see that there's this inferred energy landscape. And the energy landscape derived using this approach is very similar to that from the previous method. They're roughly linear, and the slopes are pointing in opposite directions, just as we would have predicted, where the most favorable for one is on the opposite side as the other. But we do notice a little bit of a difference, that these two estimates of the energy landscape are a little bit different in the way that the agarose tissues, the no ECM tissues, are sorting more strongly than expected from cell mechanical energy predictions alone. So if we only look at the cells sticking to each other, it's not predicting as strong as an effect as we actually observed and would infer from the actual distributions. And of course, when you're a PhD student and you've found something like this, you're like, oh God, my entire framework is screwed. Everything is wrong. Well, actually, no. We realized is that, okay, the experiment that's going to make or break this model is that we have to show that this activity term is lower. And very fortunately, / this is why I got to graduate, we did end up finding that there's this activity term that in the Boltzmann equation for a gas, it would be like a temperature. But in tissues, it's not temperature. It's going to be sort of this effective temperature of an active matter system, which represents the random undirected motion of cells. That cells as active and metabolizing particles are able to move around sort of randomly. And we can quantify this by making organoids of only one cell type. So since only one cell type is present, we can observe intrinsic cell motion that's independent of any sorting behavior. So this is just how much cells are going to be able to move around and exchange neighbors with each other. And so qualitatively from these movies, we can see that these organoids are quite different in the amount of neighbor exchange. That if you have an organoid that's sort of floating in suspension, there's very little exchange of cells. And we can quantify this using sort of a borrowed technique from active matter, where by taking the random active fluctuations or the random active fluctuations of the system sort of manifest as this effective diffusion of the components of the system. So we're using the nuclei as sort of like a probe particle or a tracer particle to estimate the effective diffusion. And so we do indeed find that without the ECM, the activity or the diffusion is a great deal lower. We also observed that one of the two cell types, the MEPs, actually seem to be responsible for most of the activity in our system, which is a little bit of a interesting behavior. So hopefully what I've shown you is that in the model organoid system, the major underlying parameters for tissue self-organization can be isolated and quantified. That I've been able to sort of say what we're observing is sort of the combination of several different forces or features of the system. And that means that we can start to understand, say, disease-related variations as altering one or more of these parameters. So hopefully, what we wanted to do then is to test this framework in the context of breast cancer. Because as I mentioned before, we think that the structure of the breast itself can kind of act as this dynamic and self-organizing barrier to cell invasion. So we tested a panel of nearly 20 genes that are strongly associated with invasive breast cancer, and predicted that some of these oncogenes would be able to increase the risk of increase the risk of observing a structure with a green cell on the outside of the tissue. So it would just shift our edge occupancy higher. And so after doing the screen, we found that lentiviral transduction with an activated mutant of PI3 kinase, which is the number 2 most common dysregulation in breast cancer, leads to a rightward shift in the steady state tissue structure. And that change in tissue structural outcomes is accompanied by a change in cell mechanical adhesion to the ECM with a measurable change in the predicted energy landscape. So, of course, you guys are all saying, PI3 kinase is at the top of this giant signaling cascade. It does so many different things. So how are you sure that LEP-ECM adhesion is the major contributor to this phenotype? So we decided to try to get at this question by knocking down TALEN1 with some shRNA. TALEN1 is a key adapter protein for integrins. So it basically is the intermediate between the cell's actin cytoskeleton and the integrins. So without this protein, cells are not able to use their integrins to attach to extracellular matrix. So using this knockdown, we observe these cells basically rounding up off the ECM because they can no longer really strongly adhere to it. And this returns these oncogene expressing cells to a more normal-like contact angle, with the result of normalizing the organoid's structural distribution. That reverting this mechanical phenotype alone, independent of any growth factor signaling that PI3 kinase is doing, has the effect of restoring the structural outcomes. And we also found that organization could also be rescued by treating these organoids with an inhibitor of AKT, which is one of PI3 kinase's major downstream effectors. However, it does so not by changing the energy landscape. So when we looked at these cells, they seemed just as happy to adhere to the extracellular matrix, but rather we saw a decrease in their activity. So we saw a decrease in the amount of nuclear diffusion measured from these live imaging movies that we made. So kind of in conclusion, and hopefully just on time, I learned that biological steady states can be maintained in these ordered structures by self-organizing forces, or this energy function that I described. But that this ordering force is kind of opposed in a complex system with messiness because of the dynamics of the system, that these cells are active, they're moving around, they're migrating. And this underlying kind of structural entropy that's simply a consequence of having multiple sites that cells can occupy. And changing these parameters, or changing the relative strengths of these parameters, changes how tissues self-organize. And of course, our future directions that we're very excited about exploring will be to sort of think about this in terms of the dynamics of kind of development and tissue development, that cells not only are changing position, but also changing identity and changing phenotype. And that there's going to be this really interesting dynamic reciprocity between cell phenotype, cell mechanical phenotype and its position, and then its microenvironment can maybe inform its phenotype. So this will also allow us to use the XKCD Science Tip. If you have a cool concept you need a name for, try dynamic entropy, because it's impossible to use in pejorative sense. And no one knows what entropy really is. So in a debate, you will always have the advantage. And with that, that concludes the science talk. I'd like to thank my PI, Zev Gartner, who's a really fabulous, interdisciplinary and creative thinker. My amazing co-author, Vasudha Srivastava, with whom I completed all of this work, and to senior scientist Jim Garby, who kind of pioneered the use of human mammary epithelial cells. We have some great collaborators who are working on some mouse models and computational models and some just straight up math that's related to our work. And I'd also like to thank my thesis committee and my funding sources. So thank you all for listening, and I am happy to answer your questions.
Thank you so much for a really cool talk. I do have some questions from the audience that I can read out to you. So the first one is, how do you capture those cool videos with the green and pink organoids?
So we do, all the cells are lentivirally transduced with either a GFP or an M-cherry in the cytoplasm. So that's why they're green and pink. And we use most of those videos were captured on our laser scanning confocal microscope, which is a very lovely confocal microscope. But we have this nice big incubator set up on it so that the cells are going to be held as though they were in a tissue culture incubator at the right temperature and the right CO2. So you just got to be a little bit careful, pick your laser powers so that you don't irradiate the crap out of them and they all die. It takes a little bit of tuning and care to analyze this data, but it's definitely possible.
Okay. And then I had somebody had a comment that said that they wanted to say I think the M&Ms really helped them understand entropy and statistical mechanics in a way that is digestible. And then I had a question, whether you use the M&M description for your dissertation.
I used the second slide of that where it's just, a green hexagon that could be in a different location. That's not in my thesis, but I did use it in my exit seminar because my parents came, so.
Okay. Well, I thought that the M&Ms was it was a very good way to kind of think about cells. I really enjoyed that. And then another question is, what would have happened if the energy term wasn't smaller? So like, what does that mean if the energy term wasn't smaller?
What does it mean if the energy term was not
smaller? When you were like, oh, this is the end of my PhD, that part.
Oh, I see. If that hadn't happened, I think it means that, so when you take a framework that's designed for, ideal gas and these hard particles bouncing off of each other at ridiculous speeds and ridiculous numbers, there's this sort of natural feeling okay, if this doesn't apply to multicellular tissues, then that's fine, right? It's not something that you would expect to work. So, if those experiments hadn't worked out, then, I would be like, oh, that's too bad. Maybe I can use some principles from this, but maybe it's not going to be kind of the core of my argument. So that's more of a science story strategic response than a science response.
Okay. Another question is that you mentioned that we can understand disease patterns by looking at these parameters. What kind of diseases would the strategy be most effective for?
Oh, that's a really great question. I have to think about the right way to answer. Well, I guess the one that I showed in my thesis is kind of the one that this is most directly applicable to, right? This is I kind of showed that, other studies have shown that the structure of the mammary epithelium is sort of this barrier against cancer progression. So, a significant number of people with breast cancer have this non-invasive right now diagnosis. And it's really important for patients to know how aggressively am I going to treat this non-invasive, lesion? And am I going to progress or not? Do I need surgery? Do I need radiation? Do I need chemo? And so better understanding like what are the risk factors that, cause progression of this disease, such as structural metrics like this, could be really important in, improving patient care. But I think that it's possible that this kind of thinking, since it's sort of more universal, like I'm using terms like energy or favorability that don't really necessarily need to be about cell sorting in the mammary gland. It could be about like buckling in, say, like the colon, for example, where, certain kinds of like colon carcinomas are associated with like a buckling change in the morphology. So there could be a number of different like mechanical energy gradients that could be described using a system like this that help describe transitions to structures that are, have worse prognoses for patients.
And so what are the challenges we face in using the clinical applications for this research?
Challenges to clinical applications for this research. I think that clinical things in the clinic need to be like relatively easy, right? So easy to access, like can we do this with maybe a fine needle aspirate? Like can I do this without actually taking a bunch of tissue out of a person? Can I do this using just the technology that's available within like any average hospital instead of in like a high-tech research lab? Those kinds of things are more like engineering questions, right? Like if we find, some kind of mechanical parameter that's really important, then it's sort of the next step then becomes sort of a classical like mechanical or biomedical engineering question of, okay, how do I build a device that can do this, relatively cheaply, and like kind of point of care diagnostics kind of question. So I think that's sort of one aspect of the translational hurdles that this kind of research has to go over in order to become useful to the clinic.
And then I have another question. What advice do you have for budding scientists who are interested in your field?
Budding scientists who are interested in this field. I think that really depends on like what stage you are, like specifically, like if you're a high school student or an undergraduate student, then like kind of the most important things are to work hard in school, like try to get some research experience of some kind. Even in high school, it might be possible to like kind of become like a high school intern at a local college, or even like through various programs that allow like high school interns to kind of do mini research projects in labs like ours. If you are maybe like thinking about applying to grad school soon, and are like kind of thinking about like what labs am I going to apply to, like what are the best programs to get into to do this kind of research, I feel that bioengineering specifically tends to have like this very like big tent approach, and that we really do like welcome people from all kinds of backgrounds, because bioengineering is itself a very interdisciplinary field and really benefits from having all these different perspectives. So if you're kind of interested in bioengineering, but you didn't do your bioengineering major, that's fine. We're happy to have like physicists, pure biology, stem cell biology, all kinds of people all bring something to the table in a bioengineering department.
So how would you describe what exactly is like your sub area in bioengineering? Because you don't only do the cell and tissue stuff, you also do the statistical stuff, you do the programming, there's MATLAB, you have mechanobiology in there. So how would you describe what exactly is your subfield in there?
That's a strategic question. Depending on who I'm talking to and whether I want them to hire me for that position, I will change what I describe myself as. But I usually say that I'm a tissue engineer with like 3D cell culture experience and like a lot of like imaging and computational background.
Okay, excellent. I think that might be all the questions that I have right now. Is there anything else you'd like to say to our attendees before I close out the session?
Oh, no, just thank you guys all for coming. I know this is like the last talk in a long day.
Yes, and thank you so much for giving this very awesome talk at a very late time. So I suppose it might be
Oh,
six.
this is like 6:40 in California. We're fine.
Yeah, it's like almost 10 for me, and Europe is asleep. Asia is also asleep. So thank you so much for giving this talk. It was very insightful. It was very different sort of tissue engineering from what I do. So I thought it was very, very cool stuff. And thank you so much for this. And I hope you enjoy the rest of your evening.
Thanks so much. Take care of yourself.
Thank you.